Welcome to the University of Rwanda, College of Science and Technology, School of Science, Chemistry Department. We are going to share a module called Recombinant DNA and Technology. I am Dr. NDAYAMBAJE, and this is my contact. If you have a question or concerns, just you may contact me. We are going just to have introduction to DNA and recombinant DNA technology, application of recombinant DNA technology, and then after we have just a summary. As introduction on DNA, this is the deoxynucleic acid. It's having two kind of components, which is a pyrimidine and the purine. This one are a component of a DNA, where pyrimidine is having three sequence nucleotides. One is cytosine, another one is a thymine, and we have also uracil. And also we are having a purine with the two sequence nucleotide, guanine and adenine. These sequences are bases pairs like AT and GC. Those are making a DNA. And before we start the recombinant DNA technology, we have to know what is a DNA and how each is following in the DNA sequence. About how we are making a recombinant DNA and technology, just we have different steps to make a recombinant DNA technology. Someone may ask what is a recombinant DNA technology. It's just to make a new organism, which is having a new properties. Just we have almost 5 steps for making a recombinant DNA technology. The first one is isolation of DNA. This DNA, you can get it from a wild type strain. It's where bioinformatics is applied, where just you can search from online and you search the gene you want. Then you can isolate that gene from wild type strain. Then you can cut that gene using restriction enzymes. These enzymes just you can buy it from the market. There are companies which are making this enzyme. After cutting this enzyme, when you are cutting DNA, you can also cut a vector. Then you can ligate, or you can just insert that gene into the vector. And then after that, you are forming a recombinant. A recombinant means it's a gene and a vector. After that, you can do linearization, because normally it's a cycle. You can make it linear, then you can transform it. Transform it means you can just insert into a new strain. That new strain, we call it a transformant, or you can call it GMO. We are familiar with the GMO. It means you have modified that organism. You call it genetically modified organism. After that, you can do expression. Expression means you can produce your products to get a product, what you want. Maybe to sell a product on the market. You know, when you want just to sell your product at the high quantity, you have to do recombinant. Otherwise, you cannot be able to produce a huge amount of the products. The application of cloning technology, mostly we are applying this recombinant in the genetic mapping, where you can just identify the locus of genes and the distance between the genes. And also you can use a recombinant in the DNA sequencing to know the sequence of nucleotides of your gene you are going to use. And also you can know the mutations, where the mutation has occurred in your sequence nucleotide or your gene or your DNA. And then we can use this recombinant in a transformation just to check the complete change in the appearance. And also it is applied in genetic engineering when you are manipulating of genetic materials, and also genome sequence, and to know the order of sequencing nucleotide within a DNA. And this was introduction on recombinant DNA technology. As a summary, as I have said, just you see, you can isolate your gene of interest. You isolate from a foreign DNA, from a wild type strain. Then you cut using restriction enzymes. Then only you can also cut your vector. A vector, it's a plasmid, it's a vector. After cutting, you can insert this one into this one. You form what we call a recombinant. This recombinant, then you can just do linearization. It means to make it linear. Then you can insert into a new bacteria. And we are forming transformation. After transformation, then we can just check whether you have a transformed. We call it a selection. Then only you can move on purifications, just to get your new product. After that, maybe if this was an introduction, if you need more information, because next time we will just go into details about the different steps in cloning technology, you can just read it, and then we could go into detail for the next time. And then some videos you may have, if you have a time, you can read these videos to have more information. Thank you very much.